Hello, and welcome once again to Tuesday live streams. So today we are talking about DNA, RNA, ATP, structure and function. And as normal, we are going to, I'm not going to read all of this stuff out, but you probably know the score, caps for questions, put topic requests in, and please subscribe, like, gossip, put your notification bell on, all of those things that might encourage other people to find this video and to use it. Okay, and I've just realized, having done this, I just need to, sorry, that was my mistake on my little bit of technology, because here am I trying to do this on my own because I don't have my normal help. Anyway, so please do gossip this channel. I want people to be able to do A-level biology. So in terms of RNA, ATP and DNA structure and function, we're going to be talking about the basic structure that is foundational to all three molecules. And we're also going to talk about how each one is different. And then in terms of each molecule's structure, we're going to talk about the individual aspects of the structure and how those aspects relate to the function of the molecule as a whole. And before we start proper, let me just say welcome to all of those of you in live land, but also welcome to all of you who are joining us kind of on catch up, so to speak. I hope this is useful for you. DNA, RNA and ATP. Here is the basic structure of all three molecules. We have a pentose sugar. We have a nitrogenous base, an organic nitrogenous base, because just as the pentose sugar is, this nitrogenous base is based on the carbon atom. And then finally, we have a phosphate group, the only bit of this whole structure that isn't based on carbon. It's just phosphorus and oxygen. This you might recognize as being a nucleotide, and it is. It is the monomer that goes to make up the polymers that are DNA and RNA. It is also the basis of ATP structure. So let's go through them all. Here we have DNA, deoxyribonucleic acid. And it is called deoxyribonucleic acid because the pentose sugar, notice I've just made it a slightly different color, is deoxyribose. It's actually the ribose sugar with an oxygen missing. Don't get hung up on that. The bases that DNA includes are adenine, thymine, cytosine, and guanine, and it has the phosphate group. RNA, notice I've shaded the pentose sugar a slightly different color because here the pentose sugar is ribose. Very similar to deoxyribose, happens to have an extra oxygen. And notice again that I have shaded the bases slightly different color because the bases involved in RNA's molecule are adenine, uracil, cytosine and guanine. There is no thymine in RNA. These two, we're going to talk about in a minute, are polymers. They're made up of lots of these individual nucleotides. ATP, on the other hand, is a single molecule as is represented here. Notice that the sugar is the same as Notice that the sugar is the same as RNA, it's ribose. There are 3 phosphate groups, and the only base that is involved in ATP structure is the base adenine. Notice that it is called adenosine triphosphate, and that's because adenine and ribose together make adenosine. You need to make sure that you don't get the name adenosine and adenine muddled up, and you never call the base in ATP adenosine, it's adenine. Hopefully that's reasonably clear. So now let's go on to talk about in more detail the structure and the function of DNA. So here is the monomer of DNA, and many of them combine to make the polymer DNA, a short section of which I'm showing here. Each individual nucleotide, notice with each, with one or of four of the bases that are involved, are joined together by phosphodiester bonds. And the base pairs, the complementary base pairs, notice that adenine always pairs with thymine and guanine always pairs with cytosine. They are joined by bonds. Notice these are dashed lines because they are simply forces of attraction. And also notice that there are two bonds between adenine and thymine, and there are three bonds between guanine and cytosine. OCR, you need to know the number of hydrogen bonds between each of the base pairs. AQA, I've never seen a question on it. The rest of the specifications, you're going to have to check some of your exam questions to see whether it is something that gives you a mark point. So DNA is a two-stranded polynucleotide. Each strand is a polynucleotide. Many of these monomers join together. And the two strands, and now I can't remember what this says. Oh yeah, the two strands are anti-parallel. They actually go in different directions. Again, this is a detail that OCR absolutely have to know about. AQA, again, I've never seen it as an answer in an exam question. But look at the way the phosphate groups are orientated. They kind of point upwards on this strand and they point downwards on this strand. The strands go in opposite directions. And if we're talking about it on a more chemical basis, it's because of the carbons that the phosphates are joined to. Because the carbon atoms in the ribose sugar are carbon atom 1 here, 2, 3, 4, and 5 here. So the phosphate is joined to the 5 carbon atom, 5 prime, it's called, carbon in ribose here, and the 3 prime here. So this strand is called the 5-3 strand. Whereas this strand here, they actually from top to bottom go in the opposite direction. It is the 3-5 strand. And finally, this whole molecule, which is kind of like a ladder, forms a double helix. And that helix you see as you look down the molecule from above. And the helix is because of the sugar phosphate backbones twining round each other. So now let's talk about the structure and function of DNA and how aspects of DNA structure lead to its function. The overall function of DNA is both the storage and the accurate inheritance, the accurate passing on of genetic information from one organism to the next generation. This accurate inheritance involves replication. DNA is the only molecule that can self-replicate. It's a remarkable feature, and it's because of its structure. So, aspects of the structure. Number one, it is very long, and therefore it holds vast amounts of information storage. Number two, it has complementary base pairs, and that leads directly to accurate replication because each strand acts as a template for the new strand being made. Hydrogen bonds. Individually, hydrogen bonds are very weak. They're not chemical bonds, and therefore they are easily broken by the enzyme DNA helicase that you're likely to remember from your DNA replication. But because there are millions of them in a single DNA molecule, many of them make the molecule strong or stable. It means that the DNA doesn't disintegrate, it doesn't decompose easily. And finally, the sugar phosphate backbone provides this double helix sugar phosphate backbone, provides both chemical and physical protection of the bases. And the bases are the crucial part of the DNA because the sequence of the bases, their order, codes for the individual amino acids and puts those amino acids in the correct order for an individual protein to be made. Because DNA, in fact, that is absolutely another function of DNA, DNA codes for proteins and therefore codes for every organism's characteristics, because each characteristic is determined by one or more proteins. So let's move on to RNA. RNA is a single-stranded polynucleotide, and you will be most familiar with this in the form of mRNA. Messenger RNA, which carries the message of DNA, of the DNA code from the nucleus from the nucleus to the ribosome, which is in the cytoplasm. Messenger RNA is single-stranded. That's reasonably apparent here. But it is a much smaller molecule than the vast length of the DNA molecule, relatively. And that's because messenger RNA is only formed for each individual gene, and therefore it's the length of a particular gene. And that means that it is a small enough molecule to exit the nucleus via a nuclear pore. DNA, on the other hand, is simply too vast in order to be able to do that. It also has a complementary base sequence, and that means that it is able to pass on the code from the DNA in the nucleus to the ribosome, and it is also able to match up with transfer RNAs, which we'll talk about in a minute, and the transfer RNA's anticodons, which means that amino acids are sequenced in the right order when a protein is made. Second RNA that we need to talk about is transfer RNA. And I like to think of transfer RNA as a molecular carrier bag, because its job is to transfer or carry specific amino acids to the ribosome so that the ribosome can bond them in the order that has been coded for by the mRNA. Notice that tRNA, like mRNA, is single-stranded, but because it loops around itself and in various places forms hydrogen bonds, it kind of looks double-stranded in places, but it isn't. It is a single-stranded RNA, but has a very specific length of a molecule and a very specific shape. Up at this end of the molecule, the amino acid is being carried, specific amino acid. And at this end, there is a equally specific anticodon. And that is complementary. This is made up of 3 bases, and it's complementary to 3 bases on the mRNA, and that's called a codon. And by matching the anticodons on the tRNA to the codons on the mRNA, it means that the amino acids are bonded together by the ribosome in the sequence that is determined by the mRNA and by extension, the DNA in the nucleus. And that is what I have written here, because the anticodons are complementary to the mRNA codon. The final type of RNA that you need to know about is ribosomal RNA, and all specifications need to know this. Very little that you need to know. You need to know that ribosomal RNA plus proteins make the ribosome. So back to our final molecule, we're nearly at the end, and that is ATP. ATP has an entirely different function. It is not a polymer like RNA and DNA. It is a single molecule, and it has a very different job. It is the immediate energy source. Oh, hold on, I've just spotted a question. tRNA molecules from hydrogen bonds, if the hold on. All right. tRNA molecules form hydrogen bonds if the bases go on the outside. Oh, no, right. Okay. These, right, let me just go back. Right. These, the hydrogen bonds that are here on the tRNA molecule are formed because the bases are internal. They're on the inside here. The only place where the bases somehow, don't ask me how, flip round are on the outside, are at the anticodon. My understanding is that everywhere else around the base, excuse me, they are on the inside of the molecule, so to speak. And in these particular places, hydrogen bonds form, stabilizing the tRNA molecule. Excuse me. I hope that clarifies that. So ATP, the immediate energy source. Really useful because it provides energy. No, let's say that again. It's really useful because a small amount, a fixed amount of energy, thank you, Julian, is released each time this terminal bond is broken. And the reason why ATP is incredibly useful is, number one, it is soluble, and that means it can diffuse around the cell. Interestingly, it can't diffuse outside of the cell. And that means, of course, there has to be a constant production of ATP inside every cell via the process of respiration for the cell to survive, because it constantly needs ATP. Because only a single bond is broken in ATP, this leads to three very useful properties. Number one, energy is easily released. It's a single bond being broken. Two, it is released in small fixed amounts. And finally, it can easily be regenerated because it is only a single bond being remade. Worth pointing out here, this is the word to use in an exam. Do not ever talk about energy being produced or made, because can you notice that if you say energy is produced, you're saying that it is a product of a chemical reaction. And that is simply not true. It is released as a result of the chemical reaction. Can I zoom in the screen? Do you know, I'm going to be really honest. I'm really sorry, Mohammed. I can't zoom in the screen right now. Apologies for that. May need to replay it on a larger screen. But I have pretty much come to the end, because that is hopefully a useful comparison of the structure and function of the RNA, the DNA, and the ATP molecules. How they are related, because they are all based on nucleotides, but how they are different because either the sugars or the bases or the number of phosphate groups in that nucleotide are different. I am hoping that that has clarified for you how those molecules are different and how their structure leads to their function. I'm just going to hang on for a few seconds now, just in case there are any questions, but otherwise I'm going to let you get on with the rest of your evening and leave this up on YouTube so that more can watch it. Okay, you are very lovely, Vanessa and Mohammed and Hambop. Thank you for the thank yous. I really appreciate it. Each of these videos does take me quite a long time to make, and I just really appreciate that you're appreciating the work that goes into them. Really appreciate your thanks, Mimi and Salisha. And I will see you on Thursday when we are going to be talking about, food tests and how non-reducing and reducing sugar food tests work, and starch and proteins and lipids. I'm working my way through those lists of techniques and skills that you need to know about for the practical aspect of the course, and all of those things you need to know about for the exams, because all the questions are in the exams, obviously. All right, I'm loving the fact that some of you are looking forward to that, and hopefully I will see you on Thursday. Bye-bye.